Okay, so during the previous session, we have looked at the structure of membrane proteins, and we talked about the distribution of amino acids and how the proteins are associated with the membranes. We talked about the integral membrane proteins. These are the proteins which are embedded in the membrane, and these proteins, they contain a region of hydrophobic amino acids on them. That portion of hydrophobic amino acids is inserted into the membrane. So through this membrane insertion, these proteins, they are associated with the membrane. And also we've talked about this earlier that some of these proteins, they pass the membrane only once, but others, they pass the membrane several times. So now we're going to look at the process of insertion of proteins into the membrane. You would have some knowledge from cell biology course about this process. Here we're going to quickly look at the mechanism that how proteins are inserted into the membrane. As we know that protein synthesis takes place on the ribosomes. Ribosomes are present in the cytoplasm of the cell. Those proteins which need to work in the cytoplasm, they are synthesized by free ribosomes. And after synthesis, these proteins are going to stay within the cytoplasm. However, not all proteins work in the cytoplasm. Several of the proteins, they need to work at other locations. For example, some proteins are localized in the cell organelles. Some proteins are embedded in the membrane of those organelles. Some of the proteins, they're secreted out of the cell, while other proteins, they're embedded in the cell membrane. So a variety of proteins have to work at different cellular locations, or sometimes outside the cell as well. For example, the proteins which form the nuclear pore, they are embedded in the membrane of the nucleus. The hydrolytic enzymes of the lysosome, they are present inside the lysosome, and therefore they have to be transported within those lysosomes. Some proteins like secreted hormones, for example insulin, they're secreted out of the cell, while many other proteins like the cell surface receptors, they're embedded in the cell membrane. So different proteins, they need to go at different locations to carry out their physiological function. This is mainly mediated by the ER-Golgi pathway. The first step is the insertion of proteins into the endoplasmic reticulum. Some of the proteins are soluble, so they are transported within the lumen of the endoplasmic reticulum, while other proteins, which are membrane proteins, they are embedded in the membrane of the endoplasmic reticulum. And then, when these proteins are transferred to the Golgi apparatus sorts these proteins, modifies them, and then eventually transport them in form of vesicles to their destination. This is a schematic representation of the process. The ribosome is shown here in gray color, and in blue color you can see the messenger RNA. The ribosome binds to the messenger RNA and starts the process of translation. As soon as the protein synthesis starts and a few amino acids are incorporated into the protein chain, the synthesis of protein is paused by the signal recognition particle. Those proteins which need to work at a certain destination other than the cytoplasm, they have a specific sequence of amino acids on them. This sequence of amino acid corresponds to the sequence of the codons which are present on this messenger RNA. When the ribosome starts translating a messenger RNA, it first translates that particular sequence of amino acids, which is typically called as the signal sequence, or sometimes called as the signal peptide. There are different types of signal sequences for various different cellular locations. For example, the proteins which are to go to the nucleus will have a different signal sequence. The proteins which have to be secreted out of the cell, they have another type of a secretions signal sequence on them. Similarly, those proteins which need to work at other cellular locations, they have a typical signal sequence on them. The proteins which need to work in the cytoplasm, they do not have any signal sequence on them. So if it is a protein which contains a signal sequence, so the signal peptide is the first part of the protein to be produced by the ribosome. Now there are these substances which are called as the signal recognition particle, abbreviated as SRP. SRP is a complex which recognizes and binds to the signal sequence. The SRP particles are free in the cytoplasm of the cell, and they keep on searching newly synthesized proteins for the presence of signal peptides. So if the SRP recognize a signal sequence on a protein synthesizes ribosome, so the SRP binds to it and it pauses the process of translation. SRP has a receptor on the surface of the endoplasmic reticulum. So the receptor is diagrammatically represented here. Now, when SRP is complex with a ribosome, it brings the entire complex to its receptor and binds here. So in this way, the entire complex, including the ribosome, messenger RNA, and partly synthesized protein, the entire complex is brought to the surface of the endoplasmic reticulum. Then, in an energy-dependent mechanism, the signal peptide is inserted into through this translocation pore. When the job is done, the SRP is released, and now the process of protein synthesis resumes. So now the ribosome will keep on translating the protein as usual, and that protein will be transported into the lumen of the endoplasmic reticulum, which is diagrammatically represented here. Many a times, the signal peptide which is present on the protein, it is removed by a specific enzyme called signal peptidase. So the signal peptidase breaks down this peptide bond here and releases the signal sequence. And hence, rest of the protein is the mature protein. Now, several of the proteins, they are soluble, so they are completely transferred into the lumen of the endoplasmic reticulum. However, those proteins which are membrane embedded proteins, they have a particular hydrophobic region in them. So that portion is inserted into the membrane, and now vesicles, when they bud off from the endoplasmic reticulum, this protein will be bound to the membrane of those vesicles. Eventually the vesicles fuse with the Golgi, so the protein will be inserted into the membrane of the Golgi, where it will undergo modification and further sorting. After sorting, these proteins will be packaged in the membrane of the vesicle from the Golgi and then will be transported to its particular destination. Okay, so this animation shows the process that how proteins are brought to the endoplasmic reticulum. In this representation, you see a molecule of messenger RNA here, and these structures are the ribosomes. So this is a ribosome bound to by a messenger RNA, and this is the signal recognition particle. The ribosome starts protein synthesis, so this is the newly synthesized chain of protein. The signal recognition particle, if it recognizes this given sequence as a signal sequence, it will bind to it, and it becomes activated. Now, the signal peptide binds to the signal recognition particle. This is showing the membrane of the endoplasmic reticulum. This is a receptor for the signal recognition particle associated with a pore in the membrane. This pore is sometimes called as the translocation pore. So the signal recognition particle brings the entire complex to the membrane of the endoplasmic reticulum by binding to its receptor. Then, in an energy dependent process, the protein is attached to the translocon. Later, the signal recognition particle dissociates, and the ribosome, along with the messenger RNA and the protein, is brought to the membrane. And through this translocation pore, the protein is going to be inserted into the through the membrane of the endoplasmic reticulum inside its lumen. So that's another animation showing the same process. So the messenger RNA is shown in blue color here, and these structures are the ribosomes which are attached to this messenger RNA. So some they're transported into the lumen of the endoplasmic reticulum, while others are membrane embedded, so they are so they're partially transported across the membrane of the endoplasmic reticulum, and hence they are inserted in the membrane. The process is similar as that when protein synthesis starts, the proteins which are to be transferred to a certain destination have a signal sequence on them. And the signal recognition particle, which is shown here, it binds to this signal sequence and translocates the entire complex to the membrane of the endoplasmic reticulum. Later, the SRP dissociates. So now the protein synthesis resumes, and the protein is going to be transported through this translocation pore into the lumen of the endoplasmic reticulum. There's this enzyme called the signal peptidase. These enzymes remove the signal sequence and produce the free protein in the lumen of the endoplasmic reticulum. Some proteins, however, are embedded in the membrane of the endoplasmic reticulum. These proteins have an inherent signal on them. That signal is important for the transfer of a particular part of the protein into the membrane. So the protein is going to be transferred through this translocation pore until it encounters a stop transfer signal. At that point, the transfer of the protein through the translocation pore stops. So some part of the protein is present inside the lumen of the ER, a part of it is embedded in the membrane, and then the remaining part is outside. Some proteins, they can have more than one start and stop transfer sequences, in which case the protein again is inserted into the membrane, and when it encounters another stop transfer signal, it is released. Then again another membrane insertion sequence and a stop transfer sequence, then a fourth one and a fifth one. In this way, those proteins which pass the membrane many times, they have multiple start and stop sequences for insertion into the membrane. In this way, these proteins are embedded in the membrane. This figure shows the typical ER-Golgi pathway that protein synthesis is taking place on the rough endoplasmic reticulum by these attached ribosomes. Later, through the vesicles, these proteins are transported to the Golgi. They keep on moving through the Golgi and eventually bud off in form of vesicles, and these vesicles they fuse with the cell membrane. Those proteins which are soluble proteins are released out of the cell, while others which are embedded in the membrane of the endoplasmic reticulum, they remain embedded in the membrane. And eventually, they are now present on the membrane of the vesicle. When this vesicle fuses with the plasma membrane of the cell, the membrane embedded proteins now become embedded in the cell membrane. This is a diagrammatic representation which shows this process. The fusion of the vesicle to the membrane is mediated by specific type of compatible proteins and lipids. So when the membrane of the vesicle fuses with a plasma membrane, these membrane embedded proteins now eventually become a part of the plasma membrane. So in this way, the proteins are inserted into the membrane of the endoplasmic reticulum, and then through vesicles they're transported to the Golgi, and then through the vesicles budding off from Golgi, they are eventually transported to the target destination. In this case, it is the membrane. So it could be the membrane of the cell, which is the plasma membrane, or it may be a membrane of some other organelle in the cell. Depending upon the type of the signal sequence, these proteins are sorted by the Golgi and then eventually through the vesicles are inserted into the target membrane. Alright, so next we are going to talk about another very important membrane structure. Those are called as the lipid raft. Lipid rafts are a type of board which the lipids use in order to travel within the membrane. Well, not literally, that was just a joke. Lipid rafts are regions of the membrane which of course are made up of lipids, but these special regions of the membrane, these are sort of patches which have low fluidity. The portion of the membrane around them has high fluidity, but these specialized patches of the membrane, these are the regions of low fluidity. Since they have low fluidity, so therefore the molecules, the particles, the proteins and other substances which are embedded into this patch, they are not able to diffuse away rapidly. So all those things which need to work together in the membrane, those proteins, those enzymes, generally which are forming complexes or which are working together, they're embedded in a small short patch of the membrane together. And because of the less fluidity in that particular area, in that small patch of the membrane, these individual molecules do not diffuse away rapidly. Such a portion, such a patch of the membrane which has low fluidity and hence the constituent molecules are going to stay together is called as a lipid raft. The fluidity is decreased by the presence of cholesterol and sphingomyelin. So this regulates the fluidity of the membrane. In some local region of the membrane, insertion of cholesterol and sphingomyelin that regulates the fluidity. So in this part of the membrane, the fluidity is less. So the proteins and the other molecules which are embedded in this short patch of the membrane, they all stay together. They do not randomly diffuse away from each other. This is typically important for multi-step processes where several membrane proteins are involved in the process. If those membrane proteins which are carrying out one particular phenomenon or a process by using a series of steps, if those proteins are randomly present in the membrane, then it is going to take longer for the process to be completed. Lipid rafts are going to hold all those proteins which are going to carry out a similar function, or they all are contributing to the same process. So the raft hold all those different proteins together, and hence the process could be taking place rapidly. This is a diagrammatic representation of the lipid rafts. So this is a lipid bilayer. The polar head groups are shown by blue circles, and the hydrophobic tails of these lipids are represented by yellow color. So this is a lipid bilayer, and you can see here that in this portion of the membrane, which is also represented here in part B, that it contains greater amount of cholesterol and sphingolipids in it. So by the presence of these particular type of lipids in the membrane, the fluidity in this particular patch of the membrane decrease. However, the lipids in the surrounding area are more mobile. they can diffuse away rapidly. However, because of the presence of these molecules, the mobility of lipids in this particular patch of the membrane is quite low. So therefore, this region has less fluid. The embedded molecules, including the proteins, since the fluidity is low, so they cannot diffuse away rapidly. Hence, the lipid raft is going to hold the molecules together. It shows the process. So here you can see that in this membrane, these are the regions which contain greater amount of cholesterol and sphingolipids in them. So this is one such patch represented here, and this is another patch which is shown here. The other regular phospholipids of the membrane are represented in yellow color. So because of this particular portion of the membrane, the fluidity in this part is less. The lipid raft contain many different types of specific proteins which carry out several important functions. There are proteins which are important for cell signaling. There are enzymes which are important for several other physiological phenomena. So by means of the lipid raft, they are held together close to each other. Proteins can join a lipid raft, or some proteins can also diffuse away. The lipid raft, they can float within the membrane as a single unit. Sometimes different lipid rafts, they may fuse with each other as well, or sometimes they may separate as well. So these structures are dynamic and the rafts can move within the membrane. They may fuse with each other or they may be segregated as well. Many a times, several proteins like receptors, they may join a lipid raft. So these related proteins like the receptors and other signaling proteins, the kinases, phosphatases, and other related proteins, they're embedded in one lipid raft. Those proteins which are going to carry out related functions and they're needed for the several steps of the same process, they generally are embedded in form of a lipid raft so that the process could be taking place rapidly. For example, this is a receptor which is shown here. That's a kinase, and then there is phosphatidylinositol. We talked about phosphatidylinositol previously that it, upon breakdown, it produces diacylglycerol and inositol triphosphate. Inositol triphosphate is then released into the cytoplasm where it opens the calcium channels and is important for the flux of calcium and hence has a role in cell signaling. So in this way, the proteins embedded in form of the lipid raft, they are able to function as one single unit. So now I'm hopeful that you will be able to understand this one. So the last thing I'm going to talk about is the role of lipids as a signaling molecule and some other important functions which they carry out. Well, we know about their two important roles that lipids are a structural constituent. Different types of lipids like sphingolipids, glycolipids, glycerophospholipids, they are the constituent of the membrane, so they have a structural role. Another important function of lipids is that they are important for storage of energy. We talked about triacylglycerols. These are the type of lipids which form fats and oils in animals and plants respectively. So they're meant for storage of energy. Yet lipids are also important for several other physiological functions. The first one in this regard is phosphatidylinositol. We have just commented about this particular phospholipid. This is a structural representation of phosphatidylinositol. This part is glycerol. This is carbon number 1 of glycerol containing a fatty acid on it. This is a saturated fatty acid shown here. This is carbon number 2 of glycerol, which is linked to an unsaturated fatty acid. And this is carbon number 3 of glycerol, which is linked to a phosphate group. This phosphate is in turn linked to inositol. Inositol is cyclohexane containing 6 hydroxyl groups on it. This is the structure of inositol shown here, that this is a cyclohexane molecule containing 6 hydroxyl groups on it. We also talked about phospholipases. There is this enzyme called phospholipase C. The enzyme phospholipase C, when it is activated in response to a signal, the enzyme breaks down this bond and releases inositol triphosphate. This is the structural representation of inositol triphosphate. The compound contains 3 phosphate groups on it, so therefore the name is inositol triphosphate. Inositol triphosphate is an important molecule with respect to signaling. Sometimes it is called as a second messenger. Second messengers are those substances, those compounds, which transfer the signal of the first messenger to the cell. The first messenger is generally a hormone or a ligand which binds to the receptor on the cell surface. Most of the time, these hormones or ligands do not enter the cell, rather they bind to specific receptors on the cell membrane. And later, the receptor produces changes in the cell which eventually release the second messenger. For example, in this case, the receptor, when bound by a ligand, activates the enzyme phospholipase C. Phospholipase C, in turn, produces the second messenger, which is inositol triphosphate. So inositol triphosphate, it then migrates within the cytoplasm of the cell, and it binds and activates the calcium channels present on the endoplasmic reticulum. Endoplasmic reticulum has several functions. One of the functions is that it is a reservoir for calcium ions. Calcium ions are needed for several important functions of the cell. For example, muscle contraction requires calcium. Movement of vesicles within the cell requires calcium. Secretion of different types of substances through the cell membrane requires calcium. So the flow of calcium within the cell is important for many different types of physiological functions. Generally, the cytoplasmic concentration of calcium is low. Calcium is stored inside the endoplasmic reticulum. There are special types of calcium channels which transport calcium from the cytoplasm into the endoplasmic reticulum. The flux of calcium into the cytoplasm is activated by inositol triphosphate. So it releases calcium from the ER into the cytoplasm of the cell, and eventually this results in secretion or other calcium mediated processes. When inositol triphosphate is removed from this molecule, the remaining part is called as diacylglycerol. So this is glycerol, carbon number 1 of glycerol, carbon 2 and carbon 3. So you can see that one fatty acyl group is attached on carbon 1, the second fatty acyl group is attached on carbon number 2. So therefore this remaining part of the molecule is diacylglycerol. Diacylglycerol is also important with respect to cell signaling. Diacylglycerol binds and activates an enzyme which is called as protein kinase C. In this figure, you can see the phosphatidyl inositol embedded in the membrane. Phosphorylated form of this phospholipid is then acted upon by phospholipase C. Phospholipase C results in releasing inositol triphosphate. This inositol triphosphate results in the release of calcium, and hence it regulates various cellular processes like activation of calcium-dependent enzymes, cell secretion, muscle contraction, and several other functions. The remaining part of the molecule is called as diacylglycerol. Diacylglycerol binds and activates an enzyme called protein kinase C. Kinases are those enzymes which phosphorylate their target proteins. So the protein kinase C, it binds to its target proteins. Most of them are enzymes, and therefore it phosphorylates them. The activity of proteins is regulated by phosphorylation dephosphorylation. Many proteins or enzymes they become activated upon phosphorylation while others become inactive upon phosphorylation. So the activity of the enzymes and the function of protein is many times is regulated by their phosphorylation and dephosphorylation. So when, so when this kinase enzyme is activated, it phosphorylates its target proteins. Hence, in this way, it is a very important type of phospholipid with respect to cell signaling and communication. The next important example is arachidonic acid. Arachidonic acid is a 20-carbon fatty acid. It is an unsaturated fatty acid. It's an omega-6 polyunsaturated fatty acid. The structure is shown here. This is a membrane phospholipid. This is glycerol, carbon 1, 2, and 3. This is the polar head group attached to carbon number 3. Carbon 1 contains a saturated fatty acid, and arachidonic acid is attached to carbon 2 of glycerol. When the enzyme phospholipase A2 is activated, it breaks down this bond and releases arachidonic acid. So this is the representation of arachidonic acid here. Arachidonic acid is later converted through enzymatic reactions into several different types of products. For example, there is a group of products called as prostaglandins. Prostaglandins are important for regulating inflammatory response. They're important for controlling muscle contraction in different parts of the body. They regulate blood pressure. They also are important for regulating fever and pain. Another important group of compounds which are produced through arachidonic acid are called as thromboxanes. Thromboxanes are important. They're produced by enzymatic reactions, and they also regulate several important physiological functions, including inflammation, blood pressure, muscle contraction, and other related processes. The third type of substances which are produced from arachidonic acid are called as leukotrienes. These are produced by several types of blood cells. So these compounds, they have an important role in mediating the inflammatory response. They're important for allergic reactions as well. In turn, the leukotrienes are also important. They bind to the target cells, they're signaling molecules, and therefore they activate cytokine production in other cells. Cytokines are also a specific type of signaling molecules which are produced by different types of immune cells. So, arachidonic acid is a precursor to several very important physiological mediators. These compounds, they regulate a lot of important processes in the body. There's a group of compounds called as non-steroidal anti-inflammatory drugs, abbreviated as NSAIDs. NSAIDs are drugs which inhibit the conversion of arachidonic acid into these mediators of inflammation. These mediators are also important for fever and pain. So therefore, the drugs like ibuprofen, the common name is brufen, and aspirin, these are the drugs which inhibit an enzyme, a key enzyme, which is important for the conversion of arachidonic acid into these physiological mediators. Hence, consuming these drugs suppresses the production of these inflammatory mediators and hence relieves the symptoms of fever and pain. The next important example are the steroid hormones. The steroid hormones are produced from cholesterol. Steroid hormones have a variety of roles to play. These are a few important examples of steroid hormones represented here. Vitamin A and D, they both are important as a vitamin. Vitamin A is needed for vision. Vitamin D is important for absorption of minerals from the gut. But in addition, they're also important as signaling molecules. They act as hormones as well. They're important for regulating several important processes in the body. They're important for controlling development and growth. They both are fat-soluble vitamins and therefore are also classified as lipids. They not only act as vitamins but also are important for cell signaling and communication. Vitamin E and vitamin K are also fat-soluble vitamins and they are classified as lipids. They are acting as a cofactor for redox reactions. They act as important antioxidants and hence they can bind to free radicals and reactive oxygen species. So, in addition to being a structural component of the membranes and also for storage, lipids are important for carrying out many other physiological functions in the bodies of living things. Lastly, we are going to take a quick look at the process of transport of lipids inside the body. The food we consume contains lipids in it, different types of lipids. Lipids are insoluble in water. The transport from the gut to other parts of the body is going to take place through the blood, and blood is an aqueous system. So therefore the lipids are not soluble in blood. The transport of lipids takes place in form of lipoproteins. Lipoproteins are complexes of proteins with lipids. One such complex is represented here in this diagram. So you can see in blue color there is a monolayer of phospholipids which encloses different types of lipids in it. So the lipids are non-polar, so the fatty acyl chains of this monolayer are facing towards the lipids and the polar head groups are projected outward. And this is a protein which is associated with this complex of lipids. So therefore, this produces a complex in which proteins are associated with vesicles of lipids. This structure is called as a lipoprotein. There are many different types of lipoproteins and they transport lipids in the body. This figure shows the process of transport of different type of lipids by using variety of lipoproteins. In this part of the figure, it shows the exogenous pathway. The exogenous pathway deals with the lipids which are coming in the body from the outside, which is by the food. And here it shows for the endogenous pathway, that is the lipids which are produced or present within the body. So let us have a quick look at this. When we consume food, the food goes into the digestive system. The lipases act upon the lipids present in the food and they digest it. Bile is also important for assimilation of fats. Eventually, these lipids are absorbed through the wall of the intestine. They assemble with proteins and they form chylomicrons. Chylomicrons are the complexes which transport lipids from the gut to other parts of the body. The enzymes called lipoprotein lipase. The lipoprotein lipase breaks down the lipids and releases fatty acids from it. These fatty acids in turn are then transported to the adipose tissue and the muscles. The adipose tissue is the fat storing tissue of the body. So these fatty acids in the adipose tissue are esterified to glycerol and hence are converted into triacylglycerol which are then stored in the adipose tissue. The remaining part is then transported to the liver. Liver has an important role with respect to distribution of lipids as well. The transport of lipids from liver to other parts of the body is mediated by several different types of lipoproteins. These include low density lipoprotein, very low density lipoprotein, and intermediate density lipoprotein. LDL, VLDL, and IDL, these are all related lipoproteins and are important for the transport of lipids from the liver to other parts of the body. HDL cholesterol, HDL is high density lipoprotein and is important because it transports cholesterol from extrahepatic tissue to the liver. LDL on the other hand transports cholesterol mainly from the liver to other parts of the body where it can be stored as well. So one important consideration is the ratio of LDL to HDL. Some people have elevated serum levels of cholesterol, and this is important risk factor for cardiovascular disorders. The LDL is mainly involved in the transport of cholesterol from the liver to the extrahepatic cells. So therefore, sometimes LDL cholesterol is referred to as bad cholesterol because when cholesterol it is transported through LDL, it increases the cholesterol concentration in circulation. Also, this cholesterol is then stored in the extrahepatic cells. HDL cholesterol, on the other hand, is sometimes called as the good cholesterol because it transports cholesterol from extrahepatic tissue to the liver. And in the liver, this cholesterol could be metabolized. One of the fates of cholesterol is the breakdown of cholesterol to produce bile acids and salts. If people have elevated cholesterol level in their serum, then they are given a drug of a group called statins. Statins are those drugs which inhibit cholesterol synthesis in the body by inhibiting an important enzyme in the pathway of cholesterol biosynthesis. There is an individual variability in the ratio of HDL to LDL. If the ratio of LDL is high in the circulation, then the probability of having elevated cholesterol level is also high. So although people advise to consume low fat diet, but still we have seen that lipids are important for many functions in the body, in the cells of the living things, and hence it is important to consume them. However, an excess of lipids or fats is bad for your health. So therefore, we must not absolutely eliminate lipids from the diet, but should take them in an appropriate amount. So therefore, both the quantity and the quality they matters. But do not be like this fellow. So with this we come to an end to this topic about lipids. Next we will talk about the other type of biomolecules which are amino acids and proteins.